Excellent. Thank you, Neil, Phil and David. We all know that building soil resilience can be the difference between success and failure for farm businesses. So it's my great honour to bring this next session into the mix, which is looking at the role of soil health. It gives me great pleasure to introduce to the stage Anne Bogle. Anne does a lot of work with us on our soil health work at AHDB. She's a senior research scientist at ADAS. And Anne, without further ado, I'll hand over to you. Thank you.
Good morning, everyone. It's really great to be here. So I'm just going to be talking and introducing the subject of soil health and what we've been doing as part of the Soil Biology and Health Partnership. So really, when we want to grow a healthy and productive and resilient crop, one which was able to withstand pest and disease, we really need to start with the soil, making sure that it's in good condition to support good establishment, good rooting, good nutrient and water uptake. So we're faced with these questions. How do we know if a soil is healthy? What drives soil health? What do we need to measure? And once we've measured those things, how do we benchmark and interpret what we find? And then finally, once we've measured and understood how healthy our soil is, what can we do to improve soil health? And this is where the Soil Biology and Health Partnership, which is funded by AHDB and BBRO, came in 5 years ago. And I've stood on this stage actually a number of times over the course of that 5 years, and we're just almost at that end point. We're just seeing the finishing line. And our remit was to improve the understanding of soil health, and particularly soil biology, but together with biology and physics and chemistry, come up with some practical measures that could be used on farm to assess soils and therefore guide management decisions. And so you've probably heard of the scorecard approach that we've developed. And this approach recognises that you can't just measure one thing when it comes to soils. You need to look at biology, chemistry and physics as a whole integrated approach. And so this approach is one that would be done on a rotational basis by farmers at the same point in the rotation, the same time and the same location in order to be able to track soil health over time. And then we use simple traffic lights in order to understand what we've measured and what further action we should take. Now, I don't intend to go into the scorecard in a lot of detail. I'm hoping that a lot of you have heard about it over the course of the 5 years. As I say, we're reaching that final goal, and there will be a video of how to do the scorecard, including all the guidance and benchmarking documentation going to be online in the next month or so. But here's just a quick summary of what is on the scorecard. So in the context of where that farm is, so the region, the rainfall in particular, and the rotation, and the topsoil character, thinking here particularly the texture, we measure a suite of properties. So on the physical side, we're looking just at a visual evaluation of soil structure in the field, the spade test, looking to score the most limiting layer. On the chemistry side, we're looking at just pH and extractable nutrients, routine soil chemistry. And then on biological side, as we're doing the spade test, we can count the worms that we find, and in a measure of organic matter. And then we can also look at microbial activity. We've got some benchmarks for some properties there. Now these measurements do fit with the Sustainable Farm Incentive Soil Standard, the measures that are asking for there. And as I say, they're things that can be done by a farmer on farm. And if you want to know more, just look out in the next month or so. There will be more coming on the GREATsoils website. But what I want to do today is show how we've been using this scorecard approach and testing it to check that it works and it is robust. but also to use it to explore what drives soil health. So we've done this at a number of long-term experimental sites, and these sites have been selected to explore those key drivers of soil health. So obviously what drives soil health is pretty much what drives human health, what we eat, what we drink, what we breathe, and the presence of any chemical toxins. So we've got 3 sites looking at different nutrient inputs and organic matter inputs. So looking at external organic matter inputs. Crop rotation is important when it comes to food source. Then we've got 2 sites looking at air and water supply. That is influenced by tillage and drainage. And then finally, we've got a long-term site looking at the chemical environment, not a specific toxin, but looking at sort of the underlying driver of pH, which obviously controls nutrient and things like heavy metal availability. So these sites provided a really good test bed for this scorecard approach, but they also provided us with a really good resource of soils with which to improve our understanding of soil biology and develop new indicators of biological functioning. So if we have a look at some of the results from these sites, so first thinking of food source. So this is Harper Adams University, where we've got a long-term site looking at repeated organic material additions. We've got similar sites at ADAS Terrington and Gleethorpe. So at this site, we have a suite of different organic materials applied for a number of different years, supplying different amounts of organic matter over that time period. So we're not looking at only the total loading of organic matter, but we're looking at different types, different qualities of organic matter, and how that might affect soil health and soil biology. Sorry. Don't want to move. There we go. Sorry. So this is the scorecard. This is what it looks like. So you've got the basic soil properties along here. And at Harper Adams, we went in twice. We went in at the very start of the programme when the site was in a grass clover ley. And then again towards the end of our experimental work last year when the site was in a cereal stubble. So in 2017, it was coming out of that grass ley, then had 3 years of arable cultivation, including potatoes. And then superimposed on that with a range of different organic materials. And I've just dipped into a couple of those. But first, I just want to draw your attention to the contrast in the scorecard between those 2 time periods. And you can also, the beauty of the scorecard is that you can immediately see by the colouring where there are issues, and then you can drill down into the numbers behind that. So you can see clearly that actually after those 3 years of arable cultivation, we've got a significant decline in our sort of scorecard compared to when it was in a grass ley, particularly when it comes to the visual evaluation of soil structure. So a deterioration in soil structure, more compact soil, and in soil biology, our earthworm numbers are considerably lower. And then, as I say, superimposed on that are these different organic materials. So the control just receives bag fertiliser, and then we've got farmyard manure and green compost applied for different timings, actually supplying different amounts of organic matter, but interestingly, improving the organic matter content to the same level relative to the control. So you can clearly see the benefits of applying those materials for boosting organic matter, and also providing a bit of a liming value as well, so boosting the soil pH, and supplying nutrients. This site was high to start with in terms of its extractable P concentration. Never received bag P in the whole duration of the trial, so 25 years. And so an inherently high value to start with. And so the flags here are showing that actually there could be a potential environmental risk of having those high P status. And obviously applying organic materials will boost that P status further. So repeated applications year on year is something that you wouldn't do necessarily in this situation. If we continue to think about food source, but here particularly crop rotation, up at SRUC in Aberdeen at Cravestone, there's a very long-term experiment where we've got an 8-course rotation in place with every stage of the rotation present in the field at the same time. Superimposed on that is a range of pH treatments, which I'll go into in a minute. But if we just think about the rotation, here's the scorecard for a number of those crops within the rotation. So we had oats, grass, wheat and potatoes. So 3 years of grass going into wheat, potatoes with oats right at the very end of that arable cropping phase. And you can see that the organic matter, actually length of time since grass has had an effect on the organic matter. So lower organic matter contents at the very end of that rotation. No impact on VES, but earthworm numbers, again, towards the end of that rotation, earthworm numbers are down, but also in the potato crop. And here also I've put one of the microbial indicators that we've got benchmarks for, the CO2 respiration burst. A fairly flat response here, but we have to remember underneath this is a range of pH treatments. So if we look at the pH scorecard for this site, so here we are, I have a range of pH levels and yes, lo and behold, we've managed to achieve those pH levels with a green flag for pH 6.5. No impact on organic matter and VES score when it comes to pH, but earthworms obviously don't like acid soils. And then this respiration burst is actually showing a nice sort of almost bell-shaped response with the optimum microbial activity being at 6.5, tailing off either side, and then pH 4.5 being particularly poor in terms of microbial activity. If we move on to look at the effect of tillage here at Loddington, now it's well known that obviously tillage will have an impact on organic matter and earthworms in particular. And what we were interested in here was, okay, if we're no-till for a considerable period of time, if we need to cultivate, say, for example, we need to control weeds or we might have an issue with soil compaction, is that going to have a detrimental effect? So a 7-year no-till field, we ploughed up 3 strips and we did the scorecard a year later. And you can see pretty good scorecard, lots of greens, but lo and behold, the earthworms are down, not surprisingly, on that 1 year of ploughing. We then went in 3 years later, did the same thing. This time, the strips have been ploughed 2 more times, so we've got 3 years of ploughing. Interestingly, the earthworm count was very similar across the two treatments. Slightly poorer structure on the plough. We went in this September, it was much drier than the September in 2018, and we think actually that probably the earthworms were hiding. So the amber flag is a review. So in that situation, we probably think, actually, maybe I should go and have another look in a month or so time when the soils are perhaps wetted up to see if we can see if this is actually a real effect, if the earthworms are there. So the scorecard works. It picks up what we would expect, those different management practices to have an impact. So it's a good practical tool, and Elizabeth will be going on how we've been using it on farms. But I wanted to just spend the last few moments thinking about how we've used the sites to explore soil biology in more depth and develop new indicators of soil biology. So here we've been looking at nematodes. Now we know nematodes as important plant parasites and vectors of disease. But actually, within the nematode community, there's representatives of the whole food web. So there are bacterial feeders, there are fungal feeders, there are things that eat each other, there are things that eat anything. So a whole range of processing communities, basically. And so actually, looking at that community structure can tell us about whether we've got a balanced community, whether the system is working well. And so we can do this by looking at different indices. You can group the nematodes into different indices according to the way they feed and according to their life cycles. So we have this enrichment index. So those are nematodes which respond rapidly. They have a very short turnover time, and they respond to nutrient inputs. And then we have a structure index. These are more long-lived nematodes that stick around in the soil. So we can plot the two against each other, and then we can apply this same principles of traffic lights to see where our soils fit. And we want to sort of end up in this sort of lower quadrant where we've basically got a relatively undisturbed community, not too enriched. And so here I've just plotted the Harper soils on that basis. So the different organic material treatments are plotted here. And you can see that for most of the organic materials, they're given a green flag. The slurry treatment, perhaps a little bit more an enriched community, not surprising. And the control treatment, a little bit more depleted and a very sort of stable community. What else can we look at when it comes to soil biology? So the other thing we've been looking at is mesofauna. So these are animals that are capable of passing through a 2 millimeter diameter hole. They are visible to the naked eye. Things like collembola and mites involved in organic matter decomposition. And so we've been identifying those to family and ideally species. This takes quite a long time and specialist knowledge with the microscope. And the work we've been doing is to see how those different mesofauna communities vary across the different sites and across the different treatments. And so here we see 3 sites. You can see that site, so soil type and what's growing and climate, has a big impact on mesofauna communities. Interestingly, here, this is the cravestone, and the different colours are pH plots. So actually no difference in the meso communities with the pH. We think this might be because they're quite mobile and the plots are quite close together, and actually they can escape those acid conditions. If we look in more detail, though, and break it down by crop type at cravestone, we can see here quite a spread of different communities as a result of the different crop types. So crop type has an effect, and as does organic matter inputs. So this is at Harper Adams, where we've got the different organic material inputs, farmyard manure here, green compost, and the other treatments there. Now, obviously, mesofauna nematodes, at the moment, that takes someone long hours with the microscope doing identification work. Not a very quick and not necessarily very cheap method of measuring soil biology. And there are actually only relatively few people in the country who can do this kind of identification. So an alternative is to use DNA. So will DNA, and so molecular markers, provide the answer to being able to understand soil biology in more depth? And here we're looking particularly at microbial DNA. This is work done by Ferrer and Lincoln as part of the partnership. So I'm really much the messenger of this, not the actual person working on it. And actually, when it comes to DNA, we found that it's really important to get the methodology right. So the microbial DNA richness and diversity is really affected by the way the soils are extracted, the sequencing platform, and the databases available to be able to link that DNA to a particular species. But over the course of the 5 years, we've developed standard protocols. And what we found is that soil management, particularly when we're looking at microbial DNA, accounts for less variation than does soil type or seasonal effects. This chart here just shows the 3 organic material sites, so 3 different soil types, Gleethorpe, Harper Adams, and Terrington, looking at the bacteria species that we found in the soils at those sites. And we saw that actually only 5% of the species that we found were common to all 3 sites. So there's a really strong soil type and location effect on bacterial communities. And really, as I say, I'm just the messenger, and we are going to be showcasing a lot of the soil biological work in the spring at a joint AHDB conference on soil biology. So if you want to find out more from someone who knows more than me about it, then do sign up for that event. So just to summarise, we've done some really good work at long-term sites and with farmers, and we found that the scorecard is a useful tool for measuring some basic soil properties. And those soil properties do show up and reflect management differences. And as I say, with regards to developing our understanding of soil biology, we're getting there, but we're not quite there yet. And with that, I'll say thank you very much.
Thank you, Anne. I don't think there is anybody that knows any more than you on the topic. Tee-hee. Thank you very much, Anne. The soil health scorecards are a brilliant tool to use, and we've been using them on the strategic and monitor farms for a couple of years now. The figures and the answers are one part of the journey. It's the discussions and drilling down deeper which really kind of give value, and it's really great to see that's going to be usable for everybody. So I'm going to hand over now to Liz Stockdale. Liz is the head of farming systems at NIAB, and she's going to really be answering the question of can we build resilience through soil health. And so, yeah, thank you for joining us, Liz. And over to you.
So I think we started the work 5 years ago, and the first question that we were always asked was, well, yeah, but soil health's all great, but actually, does it make us any money? And how does that actually work out in terms of a cost-benefit analysis? And I'm standing here to tell you today that we still don't know. So if we think about resilience, I was prepared to stand here and talk about a resilience question. Can we actually use soil health to help build resilience on farms? And how does that work? So that will tie us and lead us towards a money question, but won't take us all the way. So just to define what we're talking about, this is ecological stuff. Resistance and resilience are very tightly defined terms. Resilience is now very popular as a term. Many of you will be out there looking at that Future Farm Resilience stand, I'm sure. Resilience is one of those terms that comes from ecology and has become, well, everybody's terminology. But in that context, it still makes sense. So even in the talking of future farm resilience, the time we take to return to a pre-disturbance level following that disturbance. So how long does it take? My go-to picture for resilience is Weebles. Now, some of you are old enough to remember Weebles, many of you not. Those small toys with a sort of wobbly, what do I mean? Curved bottom, that's what I mean, because they wobble, but they never fall down. So that's the key to resilience, that ability to not resist a stress, not to necessarily be strong, but to be flexible enough to change, to have some inbuilt capacity to recover. Of course, actually then, that capacity to adapt, grow back, bounce back is key. I think it is important to say that, well, okay, if we're talking about farm resilience, and we are hugely in the industry at the moment in the context of BPS payments, actually that's linked to a lot of other kinds of resilience. And I feel a bit of a fraud given the complexity that farmers and those giving them advice on farm are doing at the moment in relation to farm resilience, in relation to business resilience, and its link to personal resilience and that real close match on farm and the real importance of those two things taken together to be talking about soil health in that context. But actually, I think it fits at the bottom of that pile. I think it is an important thing. I think there's actually some really nice evidence, and I was quite surprised when I saw it, but some social science evidence that farmers who are spending more time looking and walking in an IPM or soil health kind of context actually have higher levels of personal resilience on farm. So I'm going to make that point, even though I think that I'm a little bit of a fraud making it. A long time ago now, over 20 years ago, when Anne and I were still old in the context of soil science research, John Doran in the States put together some information to inform farmers making decisions at that point about sustainability. What changes will we need on farm to underpin more sustainable systems? That was the phraseology we were using at the time. We might now use the term resilience, and of course we might bring into this, if we really wanted to, talk of a regenerative journey and all those other sorts of things. But I don't think, or in fact I'm sure, that the things he said at the time have not changed to be different than the things we would say now. And therefore soil scientists, well, made us happy because they started to put soil on the map. They started to underpin those conversations that we were able to have with agronomists and farm managers to say, well, soil's got a real part of this sustainability piece. Because actually, if we don't minimize soil erosion, we don't conserve soil organic matter, we actually won't have production or environmental outcomes to balance. And actually, the heart of that process is around making that work most effectively in terms of the utilization of the range of resources available to us. So if we just look at perhaps what is probably our biggest change and challenge in the context of resilience, that expected climate change in the UK with our warmer, wetter winters, warmer, drier summers, yeah, maybe one day, but similar total annual rainfall, but every farmer, I think, if we ask the question, more extreme events, our rotation, and that's a fairly diverse rotation to have, but nonetheless a fairly possible rotation, a mixed systems rotation. What's going to happen on farm as a result of this climate change? Well, there will be some direct impacts on yield and productivity, but interestingly, at a UK scale, not in the southeast of England, but in the UK scale, we actually expect some potential to increase crop yield, greater variety of crops available, opportunity to build resilience and adaptation through making some small changes to rotation and management, sowing dates, varieties, those kind of things. Indirect impacts, though, are going to be huge. So that's the reduced availability of those fossil fuel-based resources, which set us a challenge against which we need to think about using things more effectively. And that's the space in which a conversation about soil health fits. The challenges are there for all types of farming systems, whether organic, regenerative, or any other. The importance here is about thinking about all these things together. Now, we did some work, SRUC and myself, for the AHDB Evidence of Farming Initiative, so looking at mitigating greenhouse gases in arable systems, building on the pillars that were there or there in the NFU Net Zero by 2040 document, hence the things down the left-hand side. Objective 1, increasing productivity. In objective 2, carbon storage, and there's an objective 3, the one that we're still waiting for all that new technology to make work better, the bioeconomy and BECCS on farm. Nonetheless, where does soil health fit? There it is, within that challenge to remove constraints to production. Because actually, if we don't remove constraints to production, we said it too, and I liked the fact we said it too. I'm using the ICM letters rather than the IPM letters, but Neil was very clear. If we don't get those constraints overcome, we can't actually make these systems work in times of greenhouse gases. But of course, if we manage to also improve our soil's organic matter, we'll be feeding into that, so those things are linked. And we feed back again. Actually, if we maintain and improve our soil health well through a number of options, we're also going to be able to improve our nutrient use efficiency. So we need to make these management recommendations in the light of climate change. Well, actually, the kind of general guidance that's about improving soil health fits there too. Minimizing tillage, increasing inputs of organic matter, varying sort of crops in time and space. And really critically understanding how each system, each individual farm is already affected by the extremes it faces. The extremes we're going to face over the next 20 years are just a little bit more of that. So we need to understand for ourselves how we've coped with our farmers, with our clients over this past extreme weather. But that adaptability of management within the constraints of markets is going to be the key to moving forward. Hence the need for not resistance, but resilience. So let's get back to soil. The Soil Biology and Soil Health Partnership, you've heard about, been key in terms of bringing information together to underpin our thoughts moving forward. Anne and I have shared slides, so I'm trying not to use ones that she's used already, but to direct you to other stuff that's coming out of the Soil Biology and Health Partnership. So the key principles in terms of soil management going forward here, the physics, chemistry, biology, now not in terms of the properties we should measure, but the targeted approaches we should take, which include getting your drainage right, getting your pH right, as Anne's shown you, as well as things around organic matter management. And that's led into the development and delivery of the Principles of Soil Management Guide for AHDB, which I'm sure is on all of your bookshelves because you got it before it went online. You've all got the printed copy. And also the leaflets that are available through that slightly bigger piece of work that included the AHDB GREATsoils team to develop those principles for soil health management in different cropping systems, recognizing that we needed to take different approaches depending on the rotational system, the livestock system that we were operating in. If you haven't found those yet, they're there on the Championing the Farmed Environment website as an access point for everybody to find. So let's just go back to our soils, understanding our land being unique, recognizing what we need to recognize, and it's very clear from the data that every soil sets its own constraints or limits. We need to take each site, and that can be not just a farm, but can be soils within a farm that behave differently. So we need to know and measure that soil character. We need to know that context against which we're then going to take our informed action using our scorecard to measure soil health. Now, we know that soil biological communities are hugely diverse. If we just map them out, that's the pH plots that Anne showed you, the bacteria, the fungal communities across the pH gradient. We can see the pH gradient effect changing, the colours change, the bars change, but what we can also see is that those communities are just, whoa, crazily coloured. There's a ridiculous amount of diversity built into those communities. And what we know from science over 20 to 25 and more years is they're also incredibly resilient. You can poke soils quite hard and they don't fall down. You can bury soils under landslips at the end of the last glaciation, dig them out literally 10,000 years later, add some green material and the microbes in the soil go, oh, thanks, and start breaking that material down. They can literally sit and wait for something interesting to turn up for that sort of length of time. So this resting state to rapid development of communities is a real characteristic of our soil biological communities. So our major functions have built-in redundancy, and both site and management drive this change. So what about resilience? So this is a diagram that I'm going to do this thing that I should point with a laser, but I'm going to go and point at the screen, that we're looking at change through time. So we start in November, move through the cycle. So what we're looking at is to what extent those communities are affected by the change. So we see a difference just in the kind of community we've got. This is data from Australia, between a situation where stubble was retained in the system, higher organic matter inputs, and stubble had all been burnt in the system. So we've got just very different communities. But the key reason I'm showing you that graph, and I'm not going into any of the stats that underpin it, is that we've also got a difference in the resilience. Look how much smaller that loop is than how much that community changes. So what we've got here is added resilience as a result of adding organic matter to this system and improving its soil health. So we've been working with farmer groups across the UK to develop the scorecard approach. So if it doesn't work, I'm blaming all the farmers we worked with. The key for that was actually what Richard was saying as he introduced me, was that actually the scorecard's been important, not just for being a scorecard, but for promoting understanding and discussion. It's been really important for thinking about, well, what do my numbers mean and where can I go and what impact has my management had? And as a consequence, the scorecard has really helped farmers to engage with those numbers and turn them into practical outcomes. So we've got 321 scorecards. Sounds like an awful lot, but actually, sorry, Amanda, who's somewhere in the audience, if you take those to even the most complicated statistics, because of the overriding driver that is sight, what you find is that it's really tricky to draw out any general principles or patterns. We've got them across heavy, medium, sandy and light soils, so we can have a look at the impact of soil texture. We've got lots of different rotations and lots of different things going on. And this is what they look like on part of my crazy spreadsheet. So this is an example of a real set of scorecards. These are all rotations with late harvested crops, dominantly potatoes in the rotation. They were all assessed in cereal stubbles or cover crops so that they're at the same point in the rotation. They're shown there from the highest to the lowest organic matter. So if you look at the organic matter line, which I'll just point to as this right-hand side, we've got the biggest number going to the lowest number. That doesn't mean they go from green to red at the bottom, because actually, of course, our benchmarks take account of soil texture and climate. So we've got that amber one turning up in the top row. And we've also got that range of pH and nutrient levels. But in general, you know what? There's not a lot of red there. Soils are not in bad condition in the UK, but actually each one of the sites has the potential to show some improvement, though there was one farm, who I'm not ever going to tell who it is, who had all green. It's just that. So just zooming in fraction more to see that data and also to do the next step of looking. So we talked about the idea that we use the scorecard to look with green and then drill down a bit further to see, well, what are we finding out about the biological function and the resilience of this system? So if we're looking at organic matter, so going from the highest to the lowest, one of the things we can do if we have the data for clay is look at that data now as a organic carbon to clay ratio. You might have seen Steve McGrath and other people talking about that. That's data coming out of Switzerland, picking up and using those ratios. So we can do the same thing with that. So that's also now coloured green, amber, no reds in that data there. And we can also look at that microbial activity in more detail. And if we want to, we can start doing things like look at, well, the activity per unit organic matter, because actually it's about how well so we can use the numbers in lots of different ways when we go beyond our basic scorecard. So we've got here our high organic matter with quite a high organic matter to clay ratio, actually very high organic matter to clay ratio. 0.1s is what we'd expect, so 0.6 is very high. So actually that soil's got unstable organic matter. It's just accumulating organic matter, it did in the past. Old heathland soil, cultivated about 50 years ago, brought into potato production. So we've got that unstable organic matter in that soil. So that's what's telling us that that organic matter is actually quite vulnerable, not very resilient. In context, this soil, you know, my favourite soil, the one that's all green, also has a really good value for that stability of its organic matter in the system. But there is this unredeeming feature, slightly lower microbial activity, so worth a conversation about whether we're providing a regular diet of organic matter into the system. So we're able to take the data from the scorecards and start to have those conversations about what they might mean in practice, but they have to be informed conversations. They're best done in local groups, across people, with people who know and understand what that data means. That'll be agronomists, discussion groups on farm and so on. Of course, all this does fit nicely into underpinning our improved grassland soil standards, our improved arable and hort standards. So this idea of basing and linking our soil health review to this bigger soil risk assessment, those risks of runoff and erosion, those things that many of you will remember from the old soil management plan days, now reinvigorated, I think, usefully to give us some guidance on farm. With each year, this idea of 20% of the land, 1 in 5, sampling on a rotational basis to add these indicators. So what do I want to say at the end of all that? Actually, better soil health does mean that farms can get the most out of the rest of the stuff they're doing. So in that context, better soil health underpins the opportunities on farm to take on board other systems. Without good soil health, with a rubbish pH, I've been on the farm of the chairman of BASE, who had me sampling because his oilseed rape crop had grown really badly and he wanted to complain to the seed merchant. We we're doing soil health scorecard work and got the data back. And I suggested that perhaps the pH of 5.5 wasn't really helping the oilseed rape very much, and perhaps a focus on liming alongside his other practices that were brilliantly improving some of his other indicators might actually get his soil back in balance. If we don't get the whole soil working, actually we can't make best use of all those other resources we're putting in. We're really starting to make progress with this understanding of the management site interactions. We're definitely not there yet, and we know that site often overrides management. So it's the reason why if this practice works here, we might not see the same outcome there, and we need to take that into those. But we do know the principles. We also do know that those relationships between soil health, rooting, water and nutrient efficiency do seem to hold really well. The data is pretty much beginning to come together, and anyone who's a NIAB member in the room, we've got some ongoing experiments with members' money that I can't show you that really starting to show that link very clearly, taking out the site factor, not plot experiments, but on-farm experiments. But actually in the same sets of studies, there is no simple relationship, whatever anyone tells you, between soil health and crop health and reducing fungicides. And Richard's going to throw something at me any minute now, if he hasn't done already. Thank you very much.
If I could invite you, Anne and Liz, up onto the stage. Oh, blimey, should have gone to Specsavers. Right, we've got a couple of minutes, so I'm going to quick fire these at you both, if that's okay. Let's go, Anne. Earthworms are not the be-all and end-all, and have been commonly used in the community for sorry, I'm going to step down here, I can read it. Should stronger caveats be placed on earthworm metrics? I'll go straight to them. Oh, there we go.
With the guidance, we will be saying that there's various things, but earthworms are a good indicator. You can do them while you're doing a soil spade test, so it actually doesn't take any additional effort to do an earthworm count, and we know they are linked. We've got the different groups. We know they are linked to certain soil functions, so they're worth doing. Yes, there are issues, there are soil type issues and all that kind of thing, but while you're digging a hole, why not count earthworms?
We're certainly not saying they're the be-all and end-all. What we're saying is they are one measure in a scorecard, and they are a potential useful measure alongside those other measures. There are others, but the earthworms are relatively free. They're time-consuming, but relatively low time-consuming, can be accessed by a farmer. The other biological measures, Anne showed nematodes, inverts, also show interesting things, but you're probably going to be paying £200, £300, £400 potentially to have that data analysed well for you. We don't think at the moment that's fit for purpose on farm.
Are there any areas that the scorecard doesn't bring in, let's say the subsoil drainage and things that people should be thinking about as well as this?
Yeah, we would always say that actually when you go out into the field, have a look at it, see if you can see evidence of surface erosion, see if you've got a drainage in particular areas of the field. If it is drained, check your drains, and it is worth actually digging a deeper hole if you can spare the effort and having a look at the subsoil, without a doubt.
Brilliant, thank you, Anne. Thank you, Liz. Liz has mentioned drainage, but how much is poor drainage hampering improvements to soil health and resilience, particularly in heavy soils with antiquated drainage systems?
Fixing drainage, if soils need draining, is a key to soil health. Having poorly drained soils is not going to support effective soil health. It's one thing. It's a really key thing. If a drainage system is there, it should be functioning well.
Some advocates are increasingly recommending or promoting livestock in the arable rotation. What evidence is there against reintroduction of livestock, particularly in arable rotations? Do either of you want to comment?
Against?
Well, I guess against, the simple against is if you're doing very simple metrics of things like greenhouse gases and methane, you will up your net carbon balance almost certainly by introducing ruminant livestock into an arable system at a very simple farm to farm level. May have no impact at UK scale, and the benefits you gain may be actually fantastic in terms of your ability to manage cover crops, maintain and manage organic matter. So actually, there's swings and roundabouts. There are clearly economic vets issues, accessibility to vets in the east of England. There are lots of careful weighing up that an arable farmer should do before they choose to make an introduction. But it's a site-by-site decision and a business-by-business decision.
I'll stay with you, Liz, on it's another one on resilience, the topic of resilience. How do farmers and agronomists manage soil health with prolonged periods of dry and wet weather? How do we build resilience into production systems?
So the dry and wet weather question is dominantly around water balance, and that's about pore space in the soil. So that's about having a really good balance of big pores to take water away quickly and smaller pores that are able to hold on to water well. It's not just about total pore size, but it's about that pore size distribution. I'm sorry to have to tell you, because it's the answer to nearly every question. If you add more organic matter to soils, you will improve your soil's ability to hold water in drought and lose water more quickly in over wet situations, but also maintaining soil structural stability at the surface and continuity of pores to the surface is key. And that's why sometimes some of our tillage systems end up having an interaction with that ability to hold and manage water.
Interestingly, actually, that at the Harper Adams site, the years when we saw a benefit of the higher organic matter content on yield were in drought years with spring cropping. So having higher organic matter content in the soil, improving water holding basically in those situations was in drought situations when we saw the greatest impact.
And is loss of ignition a suitable measure of soil organic matter? If not, where do we go next, as it seems to be a primary aspect?
So loss of ignition is an age-old discussion that we as soil scientists, how do you measure organic matter in soils? And so loss of ignition, yes, is perfectly fine. All we would say is make sure you use the same method of soil analysis and ideally the same lab for those repeat samplings, because you need to make sure you will get differences associated with method. If you want to measure carbon in soil, then the GNAS method is better. But for organic matter, loss of ignition is perfectly fine.
And if you do do mass measured, most labs will also give you the organic matter number as well, so you can benchmark your organic matter.
Liz, is there any research ongoing to look at the effect of nitrogen required with increasing soil organic matter, soil carbon levels, and how this affects the environmental impacts?
Bits and pieces. It's tricky because you need those sites that have those well-established and well-understood organic matter levels. The trial I referred to that I can't tell you about unless you're a NIAB member is doing that work, looking at nitrogen response and the interaction between nitrogen and fungicide response. I'm sure we'll be looking for that opportunity, especially with the increasing prices of nitrogen, to look at how we value that organic matter in that context better. But there is older data that can also be reassessed and reevaluated alongside that link to soil structure. So it's not just organic matter per se, it's organic matter by texture and also that link to soil structure.
I would also add that actually organic matter, it's not just carbon, you've got nitrogen and all the other nutrients in there. So actually you tend to boost your nitrogen supply where you're boosting your organic matter content.
Final comment from you both. Somebody's written, what comments would you have on pundits who say we only have 30 harvests left if we carry on the same way? What are your final thoughts first? Liz first.
It depends what you mean by the same way. So there are farmers who, if they carried on for the next 30 years, will improve their soils. There are almost certainly farmers who, if they carry on the same way for 30 years, will not have soil left to manage well in 30 years' time, particularly in the tropics. But it's an average number, and it was never 30, it was 60 or 100. But even then, it was an average number for 60 is the global average, 100 is potentially the UK average, looking at the worst examples of practice being continued without change. So as a number, it was founded in some sort of evidence. It doesn't apply to every farmer, and it's I would say an exaggeration not worth paying attention to, unless you're a scientist, Anne, and we can get money by saying to the funding bodies, oh look, we really must work on this topic.
It's a good strapline. I would totally concur with those thoughts. I mean, you just saw the scorecards that Liz was showing from across the farms. There was lots of greens, there was hardly any reds. So actually, our soils are not as bad nick as perhaps some of the press might think.
look, not only are we very lucky to have you both here today, but we're very lucky to have you working in our industry and driving change. And it's not just you, it's all your colleagues and your teams, and we're very grateful. So can we all thank Anne and Liz the usual way, please? For those of you here in Peterborough, we've got a short 15-minute coffee break out into the foyer. There's coffee and biscuits. If you're at home, go to the kitchen and have a coffee. Treat yourself to a biscuit, because we're having one. And we'll all be back in our seats here at 11:15. Thank you very much.